This method provides fast, temporally controlled CRISPR-Cas9 gene editing under a small molecule SHIELD-1. It also offers less off-target effects, lower cell toxicity, and also higher internally controlled gene editing when comparing it to constitutive activation of Cas9. The main advantage of this protocol is that it can be used for characterization of essential genes as well as tumor survival genes. The stabilization of DDCas9 is independent of its expression, and this allows the gene of interest to be co-expressed under the same promoter. This method can be easily applied to in vivo and in vitro studies. SHIELD-1 can also penetrate through blood-brain barrier, which making it easy also to study genes that are involved in brain development.
Evenly seed healthy HEK293 T cells at a density of 1 to 2 million cells per 10 centimeter tissue culture plate by using 10 milliliters of glucose DMEM with 10% filtered FBS. Incubate the cells in the tissue culture incubator at 5% carbon dioxide and 37 degrees Celsius for 20 hours. On the next day, confirm that the cells reached 70% confluency by bright field microscopy and that they are evenly dispersed across the plate. Change media 1 hour before the transfection with a final volume of 10 milliliters. Prepare 2 tubes with 500 microliters of warm media. Add 25 microliters of transfection reagent to tube 1 and incubate at room temperature for 5 minutes. To the other tube, add a mixture of plasmids,